Thanks, John, and hello, everyone. I would like to start by emphasizing that over the years, we have been continuously innovating and investing in our technology platform. Our first commercial product took about 40 months to go from strain to pilot plant run, and today we average less than a year. Our cost of product development has dropped by 90%, our time to market has reduced by 80%, and we have been able to do this by increasing the bandwidth of our R&D pipeline by 500% since 2012, while increasing OpEx by only 20%. Today, our goal is to walk you through the integrated Amyris R&D PD manufacturing technology platform. At Amyris, our goal is to disrupt conventional production systems that rely on destructive and non-sustainable practices and replace them with highly scalable, clean manufacturing powered by biology. Our platform enables the scientists to precisely engineer microbes like Saccharomyces cerevisiae to fermentatively convert sugar to a variety of ingredients that consumers use on a day-to-day basis. Using our technology platform, we can offer better performing ingredients at lower cost using a process that is far better for the environment. Now, in order to modify a microbe like Saccharomyces cerevisiae, which has evolved for billions of years to consume sugar to produce ethanol, we need to go through a highly iterative process where we design the strains, build the strains, test them, learn from the data, and based on that data, redesign the next set of strains. Now, biology being unpredictable, if we can only build and test a few strains at a time, we would need to go through a huge number of iterations through this pipeline, thereby increasing the timeline and investment needed for commercial success. We solve for this unpredictability using a proprietary lab-to-market operating system designed to continuously learn from previous iterations via highly optimized and automated molecular biology, analytical, and process development tools combined with machine learning algorithms and statistical models, enabling us to collect larger amounts of quality data. The final result is a fully integrated end-to-end R&D PD manufacturing solution that is built to disrupt how companies traditionally approach this problem. This integration means that today we start every project with the end in mind, and before we even start designing our first strain, we have a good understanding of our manufacturing process, our cost targets, and how we will get there. Today, we will walk you through this integrated pipeline in detail. So let's start with automated design, which is a proprietary programming package enabling us to access the chemistry in living systems and find the optimal biochemical route from sugar to the target molecule. Now, all living cells carry out a series of chemical reactions to convert sugar into a variety of molecules. Nature combines various chemical reactions into unique combinations to form routes or biochemical pathways. Hence, in order to engineer a microbe to convert sugar to a target molecule, the first step is to understand what reactions and routes we need to optimize within the microbe. At Amyris, we have built an atlas of all the living reactions that are known to occur in nature across multiple organisms, and we have invested considerable effort to understand how they are linked to each other. Now, the closest analogy that I can give you is that we have created the Google Maps for metabolic pathways. And just like we can ask for the most optimal route from, say, New York City to Boston from our mapping app, we can ask for the most carbon-efficient route from sugar to the target molecule. Now, just like our Maps app needs to keep track of one-way roads and shouldn't ask us to cross a river at a spot where there's no bridge, our computation program needs to keep track of reaction directionality, thermodynamics, and biological feasibility. The result isn't necessarily the shortest path from sugar to the target molecule, but the most efficient path, keeping in mind several biological limitations. The program will also calculate how much sugar can theoretically be converted to the product as shown here. So in this example, for every 100 grams of sugar fed, 23.8 grams are converted to the product. This number is important because it sets a biological limit on the manufacturing costs we can achieve at scale. Now, Every biological reaction is associated with a DNA sequence, and our program will collate a list of all the DNA sequences needed to compile these reactions in our microbes. If a microbe were a software program, then you need multiple lines of code or commands to specify a program that introduces new functionality into the microbe, so that instead of converting sugar to, say, ethanol, the microbe starts executing a new program to convert sugar to, say, farnesene. To date, we have verified programs for greater than 250 molecules. Now, Amyris got its start by producing terpenes, and we have expanded this expertise beyond terpenes. Our strategy has been to invest a lot of effort in building platform strains where we optimize the carbon flux to a common metabolic node that enable us to access a wide range of subsequent molecules with only one or two genetic changes. Today, we have a suite of platform strains stored in our freezers, and when we decide to go after a target molecule, we already have a platform strains with a large number of the genetic bells and whistles already incorporated into the strain for that target molecule. Now, this multicolored wheel on the slide is a chemical dendrogram, and it represents more than 500 molecules classified by their chemical functionality into 15 distinct chemical classes. We now have a panel of platform strains allowing us to access all these 15 chemical classes, encompassing more than 20 biochemical pathways. We have to date gone after more than 500 target molecules and produced more than 250 of them. So today, if a partner comes to us with a target molecule in mind, we have either attempted and produced that exact molecule or a molecule that is only one or two steps from the desired target. Now, let's come back to the analogy of a microbe being a computer program and DNA being code. Well, software engineers don't code in zeros and ones. They use programming languages. So why should biologists code in A, T, G, and C? At Amyris, we have hence developed a programming language for DNA called Genotype Specification Language, abbreviated as GSL. So a single line of code like the one shown here fully specifies a DNA command containing thousands of base pairs, and the process is nearly instantaneous and error-free. So let's wrap up the design section by comparing where we were in 2008, where every design had to be manually curated and specifying 20 designs would take a scientist 3 weeks, to today, where that same scientist can come up with 1,000 designs in 10 minutes or less. Now, everything we have discussed so far has been the in silico process, and we now need to introduce the DNA designs into a yeast strain using our automated strain engineering platform so that the microbe can convert sugar into the target molecule. Let me start here by using another analogy. Most of us are familiar with Legos. Let's say I give you 10 Lego parts and ask you to make a Lego car. Well, that's easy enough. But suppose I give you 10,000 Lego parts and ask you to make 1,000 Lego cars for me. That suddenly becomes a challenge. However, what if I provide you with a database that contains all the parts and a software package that allows you to design all the cars? And that same software package associates all the parts with all the designs and provides all the instructions to mix the parts and assemble the cars on an automated workstation. Well, we apply the same analogy to strain construction. Every DNA part has a specific function, and we have a database of greater than 200,000 DNA parts and a software package that associates the parts with the designs and computes the robotic worklist to assemble the DNA designs. Let's continue with the Legos analogy a little more. Every Lego part has a unique function, but each Lego part can be connected to another part due to the compatible ends. We apply the same concept to DNA parts, and we introduce common sequences at the ends of all our DNA parts so that when different DNA parts are mixed in a single reaction, they come together in a predictable fashion to form the large target DNA design, which is then inserted into the strain. We currently assemble about 1,500 DNA designs per cycle and start a cycle every 2 weeks. So let's wrap up the strain construction section by comparing where we were in 2008 when all our strain construction operations were manual to today where we have applied a high level of automation to the whole process, resulting in a 1,000-fold increase in throughput but with 100-fold lower costs. The next step in the lab to market pipeline is screening all the strains that have been generated. At Amyris, we generate more than 500,000 unique genotypes that result in about 7.2 million strain candidates in a year, which now need to be screened to find the strains that efficiently convert sugar to the final molecule. Think of this process as a giant funnel which sorts out the performing from the non-performing strain. At the end of the day, on average, only about 100 of the top strains are sent to fermentation per month. This screening process required a lot of investment in terms of development of proprietary statistical models and machine learning algorithms. You have to remember that the end state for our strains is performance in large fermentation tanks. But there isn't enough capacity in the world to test all our strains in fermenters. So we instead use 96-well culture plates to rapidly sort through the strains. When we first started measuring strain performance in plates and try to predict a fermenter tank performance based on individual measurements, the data is as shown on the left. On the x-axis is the performance in plates based on 4 different types of plate datasets. The y-axis shows how those same strains performed in tanks. If the correlation between plates and tanks was good, you would expect the data points to cluster closer together along the line, but instead we see a complete scattering of the points showing poor correlation. The reason for this is obvious. The conditions in a plate are quite different from those in a tank. So in effect, it's the equivalent of trying to pick a world-class marathon runner, but you can only test your athlete using a rapid 100-meter race. So what we started doing is measuring multiple data sets and combining them all in a multivariate analysis model and using statistics and machine learning to predict tank performance. We not only measure the levels of our target molecules, but a whole range of intermediates that the strains produce en route to the target molecules, as well as side products that the strains may be making. When we started implementing these models, we started observing better correlation between our predicted and observed tank yields, as shown by the data points coalescing along a straight line in the right-hand graph. Today, we develop models like these for every new molecule project that we go through. So again, in 2008, we were working on a single molecule, only ran a single assay. And today, we run more than 30 assays for more than 18 molecules on any given day at a throughput that is 100-fold higher. And more importantly, we are collecting about 100-fold more data than we used to. While we consider our platform so far to be disruptive, the next phase is where we truly differentiate ourselves from other companies in our space. And this is our experience in traversing the valley of death that all companies need to go through when scaling up their strains and lab processes. When you think about where you start in the lab at 100 microliters and where we end up at 100,000 liters or more or higher, you're talking about a billion-fold increase in scale. And the transition from each step to the next is a failure point that can derail the entire project. We have been successful at managing these transitions with a well-developed fermentation pipeline where the data quality is robust and where we are measuring the right parameters to enable success at the next scale. Our experience with process development on 13 molecules has allowed us to develop lab-scale processes that mimic manufacturing scale, such that our performance translates perfectly across various platforms, as shown by the tight correlation in this graph for real-world data, comparing our lab performance to commercial-scale performance. What this means is that we can go from a half-liter tank directly to a 200,000-liter tank with high reproducibility, thereby reducing risk and cost and speeding time to market. So again, in 2008, we used to run standard 2-liter tanks and ran one process and had experience with just one molecule. Today, we run a wide range of high-throughput fermentation and purification systems and have developed more than 50 processes and have collected data for more than 50 molecules. Let's now see how this all comes together using Roboticide M or RebM as a case study. Amyris' RebM is a great tasting, natural, non-GMO, zero-calorie sweetener with a market value of at least $2 billion. It is 200 to 400-fold sweeter than regular sugar depending on the application and formulation, and our process results in a product with no bitter aftertaste. RebM is an extremely complicated molecule to make via fermentation, and yet our team has been able to double strain yield and productivity every 6 to 9 months, and we have now surpassed our original targets. And to emphasize just what it takes to achieve success in this space, this is what it took to double strain performance every 6 to 9 months. We had to optimize 8 different enzymes, screened about 100 genes per screen, and carried out 6 separate enzyme evolution projects. On the strain side, we introduced 17 novel enzyme activities that did not previously exist in yeast, altered 53 native genes, screened more than 500,000 strains, and ran more than 1,500 fermentations to find the right manufacturing candidates. And this doesn't even include the novel fermentation and downstream purification operations that a process development team implemented to drive cost down. So, to conclude, let's reiterate what we covered today. Since 2012, we have continued to deploy our industry-leading R&D, PD, and manufacturing platforms to the commercial scale manufacturing of 13 different molecules. We have done this by continuously innovating in all facets of a fully integrated platform, allowing us to reduce product development costs and time to market. John, your thoughts on what we have communicated so far?
Thanks, Sunil. I really appreciate the overview, and I'm always in awe about the amazing power of biology, and it's John Doerr, our largest shareholder, always has a simple way to explain it. His view is, you feed these amazing bugs sugar, and they poop amazing molecules. The stuff in between is exactly what you just described, which is actually where all the magic is. And I think we're just scratching the surface. It's taken quite a bit to get here. I think a lot of times people bring up competitor names as like, oh yeah, they've got a great story and look at all the buzzwords they use. So how are you different? And everything you've just said and the metrics you just put out as to the progress we've made in lowering costs, increasing productivity and throughput, focusing on cycle time to learning, and always starting with the end in mind. Because at the end of the day, the technology only matters if it actually makes a product that makes the world better.